compartments where different reactions would be occurring. My name is Macaulay Clark. I am a final year undergraduate student at the University of the West Indies, Mona. I'm passionate about climate change, food security, and the application of biophysics to biotechnology. I'm also an artist and I love to paint. So it's a little icebreaker, but I don't think we have time for that right now. So I'll move on to the definitions. What is a cell? A cell is the base component of living organisms. Some are multicellular and some are unicellular. Then we have the definition of metabolism. The metabolism is the combined reactions occurring in an organism to ensure its functionality and survival. This would include the respiratory reactions, aerobic and anaerobic. See the message in the chat. Okay. Okay, I thought it was 9 to 9:30 and then 10:30 to 11:30. Okay, so we could go back to the this then. What is your name and what scientific topic or topics are you passionate about? And do you have any hobbies? You could open your mic and share if you'd like to, or you could type in the chat because I can see in the chat now. I saw one before. I could read that, I guess. Hi, Giovanni. Giovanni says he's 19 years old and he is interested in the biology branch of science. That's nice. Biology is fun. It's broad though. Anyone else? Hi, Shamalia. Shamalia Abrams is an invited teacher for this camp. She is in the fields of geography and biology. She says it's always been her favorite subjects. She's an aviation enthusiast and she loves climate change and sustainability actions. She has developed a deep love for the natural world and it has led her on an incredible journey of discovery. She's also 18 years old. Shania Stevens, good morning. Oh, marine biology, nice. I love marine biology. Okay, I'll move on, and anything else that's added to the chat, I'll just read.
Crystal actually has her hand up.
Oh,
so
sorry, I didn't see that.
She can go, yeah.
You could go ahead.
Oh, my name is Crystal Mitchell and I'm 17 and I want to do something in biochemistry or environmental engineering.
You could do both.
Oh, yeah. And my hobby, I guess, relating to this, I have like a environmental blog.
That's cool.
Yeah.
Nice. Debbie says biochemistry. Biochemistry is fun. Okay, I'll move on now. You could lower your hand, Crystal. It is full of headaches. All right, so we're past the definitions. Now we're on to the general synopsis of cell metabolism, well, cells to metabolism / cell metabolism. We have cell membranes, the key biomolecules, carbohydrate metabolism, fatty acid metabolism, amino acid metabolism, nucleic acid metabolism, and enzymes. I did not highlight anabolism and catabolism because I wasn't sure of the age range that would have been here today. So yes, that could be a little homework. Defining anabolism and catabolism and what reactions would be occurring on those two different types of pathways, because there are a lot of pathways within them. So we have cell membranes. To understand cell metabolism in its entirety, you have to be able to grasp how cell membranes work, how enzymes work, and how the chemistry correlates with everything, because at the core of everything, the chemistry is the most important aspect of what you'll be learning as you further in your studies in these topics. So we have the cell. If anybody could identify what type of eukaryotic cell this is. Okay. Yes, it is an animal cell. So we have the different compartments within the cell that different reactions would be taking place across the membranes, and within the compartments, you have the mitochondria, where we have the ATP production. You have the nucleus, where there's the nuclear membrane. You have the endoplasmic reticulum and the Golgi apparatus and all of that. And it's lacking the chloroplast, so you know for sure it is not a plant cell or a photosynthetic bacterial cell or archaea. And then we have this one. Since we know that one was animal and this one is plant, since I said eukaryotic cells, we have the nucleus, we have the chloroplast, identifying factor, and then you have a large vacuole, and all of these different compartments would have different reactions occurring across the membranes, and you would also have reactions occurring within the cytoplasm of the cells. Now we have cell membrane, and this part, you could see my mouse. Okay. And this part of the membrane is the extracellular fluid. So if this is a part of, say, a blood cell, this is the membrane of a blood cell, you would have the extracellular fluid, which would be the other components of the blood here, and then you have the internal components of the blood cell in the cytoplasm here, and then you'd have the membrane proteins, you have the intrinsic membrane proteins, and then you have the peripheral membrane proteins, and then you have the membrane associated proteins, which would be attached to the glycolipids. And there. And you'd also have the sorry, I said glycolipids, you'd have them attached to the phospholipids, and then you'd call them glycolipids if it's a lipid attached. What am I saying? I'm so sorry. Yeah, you have the phospholipids in the membrane, so you have the phosphate head, the lipid tails, everyone here is, well, everyone that has identified themselves should be familiar with membrane structure since you're at the age where you would have done biology, yes? Yes? I'm guessing. I'm not sure. Okay, great. Yeah, so you have the phosphate heads, you have the lipid tails, and then you have other components associated with the bilayer. And then another layer for another name for the cell membrane would also be the plasma membrane, and it's interchangeable. The same thing. Okay. So it's more detail on membrane proteins. You'll have different types of integral proteins, and then you'd have different types of peripheral proteins. So you'd have the peripheral proteins that just associate with the phosphate heads and they rest on top of it, but it wouldn't be resting, it would be chemical interactions because the proteins are made up of the amino acid monomers. So you would have interactions between the phosphate chemical properties and the amino acid chemical properties to keep it in place. So that's where the importance of chemical bonding would come into play when you're studying to understand that. And then this would be one that's attached to, this would be a lipid and like a sugar here, and then you have a protein attached at the top. This doesn't necessarily have to be a lipid. It could be cholesterol as well, or anything else that's integrated in it. But the protein is peripheral, so the protein would not be this part that's within the membrane. And then with the integral proteins, you have the anchored proteins. So this would be a lipid anchored protein. You have the protein here, and then you have the lipid here. And then this would be a glycoprotein lipid anchored. So this would be a sugar. And then you have the phospholipid, and then you have the protein. These would be, well, not all of them are channels, but this one is a transmembrane and this one is a transmembrane. These would be porins. Porins are what you call the channels, and you would have the molecules moving into and out of the cells through them by diffusion, whether it's facilitated, active, or passive. So this one would be an open porin, and this one is possibly facilitated like a ion pump or something of the sorts, but this is multi-pass. And then you would have ones that are single pass. So something that looks like this over here would be a single pass where it goes through one time. But this would have the long protein. This is one protein, and it just passes through the membrane multiple times. And this just rests on one layer. Anybody have any questions related to the membrane proteins before I move forward from the proteins? Okay, so this one. Okay, yes, everyone is fine so far. And then we have different types of membrane proteins. You have the porins, and then you have the signal molecules. I described what the porins were. The porins are channels that allow molecules into and out of the cells by diffusion, as active, passive, or facilitative transport, or osmotically through aquaporins for water transport. So water goes into and out of the cells through aquaporins. That's specific pores in the membranes for water to be transported, because water is essential for all of the reactions, since the hydrogen molecules power a lot of pumps and trigger a lot of enzyme reactions. Now we move on to biomolecules. Can anyone name the 4 most important biomolecules that would be important to metabolic activities? Giovanni says proteins, yeah, lipids, yes, proteins, nucleic acids, yes. Carbohydrates, proteins, lipids, nucleic acids, carbohydrates, yes. So for proteins and lipids, you would have to refer to them as amino acids and fatty acids, as the protein is a type of amino acid that is the polymer for the amino acids. And then you would have the monomers, you would have the dimers, and you would have oligomers. And all of those would fall within the amino acid category of that. And then lipids, you would have the different types of fatty acids, but lipids are a type of fatty acids. So yeah, biomolecules are core chemical components that sustain life, this is the typo, and ensure the survival of organisms. The 4 main biomolecules are carbohydrates, fatty acids, amino acids, and nucleic acids. These power the reactions that we will be going through. We have the metabolism of biochemicals. Does anybody know any metabolic reactions that they could list off the top of their head, and what biochemical would be associated with it? What biomolecules, sorry, supposed to be biomolecules, not biochemicals. Yes, DNA synthesis is one. Yeah, the nucleic acids. Yeah. Anyone else? Okay, that's fine if you can't remember anymore. One is enough. So we have the this is a general metabolic pathway map. There are more in-depth ones that you can look for, and they would take up the entire screen and you'd have to zoom in on them, but it's very cool. So you should look for one, and they would have all of the reactions that occur within the metabolic pathway across organisms. So we have 4 amino acids. They form a pool of nitrogen when they're digested, and then you'd have these formed in the different organisms. And you'd also have the urea cycle. For carbohydrates, you'd have glucose being broken down from the polymer that you started with, if you started with a polymer, or you'd have the conversion to glucose if it is this is happening in an organism that needs glucose to function. Say if you hadn't eaten anything for the day and you had something that contained lactose, your body would convert that to glucose. So it could go through glycolysis to form pyruvate and then ATP and all that. Acetyl-CoA and all these things. And then you'd have the lipids, the fatty acids being broken down through beta oxidation. All of this would be channeling in towards the production and utilization of ATP, and you need water and oxygen for this to occur in aerobic organisms. So since we're humans, we'd be going with aerobic. Okay, so this was a question. What happens when you eat? Do you know what happens when you eat a meal that contains all of the key biomolecules? If anyone can, let's say it had a sandwich and it had bread, it had meat. Meat would have the fatty acids and it would have the amino acids, well, the proteins and the lipids. We could go with that, and then they get broken down. And then you would have some fruits and vegetables. They would have more vitamins and all that. And it would also have nucleic acids. But yeah, so yeah, anyone wants to try and give a little map of what happens if you eat a sandwich? You could use your hand and pull on your mic and go through it if you want to. Nobody? Okay. Okay. They're broken down, Giovanni says they're broken down to monomers that our bodies can use. So proteins would be broken down into amino acids, and carbohydrates would be broken down into glucose as a series of enzyme-aided reactions. Yep, exactly. That's right. Was initially, I was supposed to stop here and make this like a write-up thing, but we're going until 10:30. And did not have the rest of something. Okay, so we have the carbohydrate metabolism. I'm sorry, we back on. Sorry about that. So we have the carbohydrate metabolism. You would eat the starch and it would be broken down to glucose at some point within the body. You could have the starch being broken down to glucose, or you could have, if your body is in a state of starvation or you're having a low glucose count, you're diabetic, essentially. Your body would break down lipids, it would break down all of the other biochemical biomolecules to form glucose. So you would have the reverse of the lipid metabolism, where you form the adipose cells. They would come out of storage and then they would be broken down into pyruvate. So see here, you have fat to acetyl-CoA, and the acetyl-CoA goes to pyruvate, and then you have gluconeogenesis, where you utilize ATP and you utilize NADH, and it goes to form glucose 6-phosphate. Glucose 6-phosphate is the only form of glucose that can go across the membrane, because the membrane protein that is associated with glucose is specific to the phosphorylated carbon molecule. So the 6th glucose is phosphorylated, and that is what the glycoprotein, that is what the sorry, not the glycoprotein, that is what the membrane protein would recognize, and it would channel the glucose into the cell as glucose 6-phosphate, and then glycolysis will occur where it is converted to pyruvate. Again, I was not sure of the ages, so I did not include the full cycle, but I will go into it. So give me a minute. Give me a minute to get that, and I'll go into it since we're going until 8:30. Okay, so I'll be reading and you can stop me and ask me questions wherever you need me to stop and ask questions. And I will share the notes with you all so you can have it, since it would be beneficial to you all to have this information, considering that you'll want to go into biology and biochemistry. Okay, so I'll be going through glycolysis now. So the glucose molecule is phosphorylated by an enzyme in okay, first let me start from it being outside of the cell membrane. The glucose is outside of the cell membrane and it is recognized by a GLUT protein. So that's a glucose recognition protein. And then it enters an hexokinase. This is in the liver. Hexokinase is stimulated by glucose, and it is inhibited when insulin okay, yes. It's inhibited when there's excess glucose 6-phosphate in the cell. So you can't have too much of the glucose in the cell. That means that there's a buildup, or that means that the end product is not being produced anymore. It's not needed anymore. So it would stop producing it because your body has a lot of, well, not just your body, cells have a tightly knit regulatory system. All cells, plant cells, bacterial cells, archaea cells, they are tightly regulated, especially by feedback regulation. So if it is you have too much glucose 6-phosphate, that means that it's not being broken down anymore. And at some point within the metabolic pathway, you don't have acetyl-CoA production by glucose needed, or it's not happening because you're diabetic or something. That doesn't necessarily have to be the case because there's a lot of interconnectedness in the pathways happening. And then after the glucose 6-phosphate is formed, this is assuming that the pathway is going smoothly and you need pyruvate to be formed to form the acetyl-CoA. You would have phosphohexase isomerizing the glucose into fructose 6-phosphate. So you have the hexose sugar being converted into the pentose fructose, but the phosphate is still on the 6 carbon. If anybody wants me to, is everybody following? I'm not sure. Is this okay? Okay. Okay, I'll share the notes after, but yeah. Where was I? Okay, yeah. So it's isomerized into fructose 6-phosphate. Then you would have phosphofructokinase 1. This converts the fructose 6-phosphate into fructose 1,6-bisphosphate. So you add in another phosphate onto the carbon 1 of fructose, and you have the phosphate on carbon 6 of the fructose. And then, so this is a polar molecule. You have the phosphate on this end and you have the phosphate on this end. And it would go 2 different pathways because you need a 3-carbon molecule to be able to form the pyruvate. So this is when the 6-carbon molecule splits into the 2 3-carbon molecules. And so far, you used ATP to form glucose 6-phosphate. You use ATP to form fructose 1,6-bisphosphate. Okay. So we're at the splitting of the 6-carbon molecule. You would have aldolase cleaving it. And you would have glyceraldehyde 3-phosphate. That's the one with the phosphate on the 6-carbon. And then you would have dihydroxyacetone phosphate, DHAP. That's the one with the phosphate on the, I'm reversed. That's the one with the phosphate on the 1st carbon. So you have one with the phosphate on carbon 1, and then you have one with the phosphate on carbon 3 in the 2 different molecules. So now at this step, you have the 2 different 3-carbon molecules. They're mirror images of each other. And now you would have triose phosphate isomerase come in to convert the DHAP into glyceraldehyde 3-phosphate because DHAP cannot continue in the glycolysis pathway. Since it's an acetone, it's not going to be able to form pyruvate like that because you need it to be still sugar related. Now you have the 2 glyceraldehyde 3-phosphates. And you would have NADH plus come in and reduce those molecules with glyceraldehyde 3-phosphate dehydrogenase. So you're removing a hydrogen molecule from each of the 2 3-carbon molecules. And you're adding a phosphate to it as well, a free inorganic phosphate. So you would have 2 polar 3-carbon molecules. So you have a phosphate on this and a phosphate on this end. And then you would call that 1,3-bisphosphoglycerate. And the next step, you use an ATP with phosphoglycerate kinase. Sorry, I need to drink some water. I'm sorry. So yes, you have the 2 polar 3-carbon molecules. That's 2,1,3-bisphosphoglycerate being converted to 3-phosphoglycerate. So you're using ATP to oxidize these molecules. Kinase is when you're using ATP to remove or add a phosphate to a molecule, and it would include the use of water as well for these reactions, since that's kind of a linked reaction more often than not. So yeah, you'll be using 2 ATP since you have 2 of the 3-carbon molecules, and you will have 2 molecules of 3-phosphoglycerate. And then you would have phosphoglycerate mutase that's moving the phosphate that's on the 3rd carbon. You remove the phosphate from the 1st carbon. So the phosphate is on the 3rd carbon. You're moving it with phosphoglycerate mutase to mutate it. So you're moving that to the 2nd carbon. So now you have phosphoglycerate, 2-phosphoglycerate, because it moved from the 2 to the 3. And then you use enolase to add, you use enolase to add a carbon group to the 2nd. Somebody said something. Okay, I thought somebody was saying something. So you would add the carbon group with the oxygen on it to the 2nd carbon between the phosphate and the phosphoglycerate. And now that is phosphoenol pyruvate because you're adding an enol group. And that's called PEP. And then you form an ATP again now by removing the phosphate from that molecule. And then you would have a pyruvate. So you formed 2 ATP at this step using pyruvate kinase from PEP. You form 2 ATP at this step with the 1,3-bisphosphoglycerate. So that's 4 ATP so far from 1 glucose molecule. You formed 2 NADH. You utilized 2 ATP. So a net, you would have how much ATP to form 2 molecules of pyruvate from 1 glucose, if anyone was following. Nobody can answer right now, that's fine. I'll be sharing the notes, and in the notes I have the chemical compositions of them and the conformation of the molecule, so you'll be able to follow it easier and answer some questions that I would put up in the Google Classroom. Somebody answered. I asked how many ATP do you have net after breaking down the one glucose molecule into two molecules of pyruvate at the end of that? Yes, yeah, two. Yeah, we used two at the beginning. Okay, great. For the regulation of glycolysis, if there's too much glucose in the blood, hexokinase recognizes the excess glucose and stimulates the enzyme to catalyze production of glucose 6-phosphate. So if glucose 6-phosphate is produced, then you would have the automatic conduction of the glycolysis pathway unless there is another regulatory step being enacted within the pathway, or unless there is something wrong with the cell. Then we have phosphoryptokinase 1. Insulin inhibits the production of fructokinase 2, 6-bisphosphate. Sorry, fructose 2,6-bisphosphate, which stimulates PFK1 to produce the fructose 1,6-bisphosphate. So if insulin is present in the cell, it would inhibit the production of fructose 2,6-bisphosphate, and fructose 2,6-bisphosphate sorry, that was excess information. I'll just leave that out because that's a little bit too much. So if there's excess ATP, NADH, or citrates in the body, you would have citrate being produced from the citric acid cycle in the last further steps from pyruvate when the pyruvate goes on towards the mitochondria. Okay. So pyruvate kinase would recognize the excess ATP and allosterically being inhibited to stop PEP synthesis. So this kind of shows the trend that the regulation of the pathways occur by too much or too little in the blood or within your cells in general. So if it is the organism isn't eating enough of one thing, or if it's eating too much of one thing, your body would react to suit to balance out reaction. So homeostasis occurs at every level within the organism, especially on the chemical level. Okay. I'm so sorry I did not get to add the pictures for the other stuff, but I will be sharing it. So what happens to the pyruvate after is that it would take other pathways. So I'm going to look for a better picture of the map and share it with y'all. So you could see the pathway map I was talking about and what you should download if it is you want a pathway map if you don't have one. I think a lot of advanced biochemistry textbooks have them as well. Anybody has anything to add or to ask whilst looking for the map to share it? Mm. I shared the I shared the map in the chat. Giovanni is asking, can scientists synthesize monomers artificially, or is it just polymers they can synthesize artificially? What are you asking exactly? You wanna put on your mic and ask like what perspective you're asking from? Because I don't want to answer you wrong. Yes, they can. Because if you look at the map, you'd see that you can reverse some pathways. So you would have the fatty acid beta oxidation, where you would utilize the fatty acids to form pyruvate and these things. But remember that glucose and amino acids and singular atoms, and these things are made up of atoms. Whether they can do it, yes, but it's a matter of people wanting to consume it and ethics and all of these things. But theoretically, it is possible. They can make amino acids and glucose as long as they're able to replicate the reactions, which they should be able to do utilizing enzyme chemistry and all that. Did it? Okay. Everyone could see the map properly. I don't know if I could upload to share it. Is everyone seeing the map that I sent? Okay, great. You're able to see it when I open it on my screen, or it's not showing and I have to share the entire screen? Okay, let me do that. So it's easier to follow. Okay, you seeing it now? It's a little blurry. Okay. Sorry, it's good. Okay. So we started with carbohydrate metabolism. Carbohydrate metabolism is where most of your studies in biochemistry would start, if not all, because the carbohydrates are the main source of energy for your brain and your body cells. And a lot of courses would be human biology related until you start to specialize more and move on to plant biochemistry once you get past your first set of advanced courses. So you'd have the metabolic pathway for sugars, and you would have the glycolysis. Glycolysis is here, is a little blurry, but glycolysis is here, and gluconeogenesis is going the opposite direction of glycolysis because it's the opposite direction, but it's not the same set of reactions and it's not the same enzymes being utilized, even though you're forming the opposite products. So you would have, so pyruvate here. I'll find a better labeled map for you all. This one isn't labeled too well. But you would have pyruvate at one of these, and then that would go back into glucose, or you would have the glucose and it goes to pyruvate. I think this one is a pyruvate because you have these other reactions when all that. And the pyruvate goes towards acetyl-CoA. Acetyl-CoA can go towards fatty acid synthesis. And then fatty acid synthesis would go towards your adipose synthesis and fat stores in your body. So you would have the fatty acids going through the specific channels in your cell membranes to get to the fat stores in your body. And you would have specific enzymes working within the fatty acid synthesis metabolism, because this is an entire metabolism in itself. Fatty acid, beta oxidation is also with fatty acid. So this is the reverse of fatty acid synthesis, as gluconeogenesis is the reverse of glycolysis. So you would have the fatty acids, well, the fats in your fat stores undergoing the conversion back into a molecule that would be able to travel through your bloodstream into the cells that need the pyruvate or the glucose, because it's being, if you follow it here, it's being oxidized back into acetyl-CoA, and all the pathways would lead back. So you have the acetyl-CoA here, and then it goes this way, you form malate, and then it goes back into glucose. or it could follow other pathways. It could go into the pentose phosphate pathway, and the pentose phosphate pathway would go into nucleic acid synthesis. And then the nucleic acid synthesis would go into DNA synthesis and RNA synthesis. And those are quite complex, but I would share the notes. I would share the notes for all of these specific pathways if it is you all want to see them in detail. But yes. And then carbon fixation and photorespiration, these would be specific. Okay, great. These would be specific to plants, but these are common in all eukaryotic cells. And then you would have them common in some prokaryotes and some archaea, but they wouldn't have the same mechanisms as those would be unicellular organisms, and they would be occurring within one compartment because it's not compartmentalized. So you would have different enzymes working as well, because you don't have the ability to store them in different places to carry out the reactions uninterrupted, because the chemicals would tend to interfere with each other and the capacity to do what it's supposed to do. Oh, and you have the urea cycle. The urea cycle also leads back to the nucleotide metabolism, or the protein metabolism, but these would break down the proteins, because the proteins would have nitrogen, and nitrogen is important in nucleotide and nucleoside synthesis. I think that is it in synopsis. Anything you wanted me to touch on again that wasn't clear, or anything that anyone wants to add? Oh, I'll also add that your metabolism is extremely important for activity in your nucleus. So your DNA replication, your transcription and translation, and all of these things rely heavily on your ability to energize the cell, because especially for transcription, when you have the addition of the poly A tail, the A's that are being added are not free flowing, they're not free flowing adenosine in the cell. You're taking the adenosine from multiple molecules of ATP. So you need a lot of ATP to be able to form that one transcript. And that one transcript could be the insulin, for the insulin that you need to be able to conduct your carbohydrate metabolism. So eating is important. Eating a well-balanced diet is important. No questions? Well, since nobody has any questions, I will make the notes available and I will set, well, I would put the practice questions in the Google Classroom. They'll mostly be like short, well, not short answer questions, which would be not essays, but yeah. As well as notes. Thank you for paying attention and asking questions and interacting. I will see you all at the next session.
Yeah, good morning, everyone. I saw that we have some